Hi all, I'm Miran Öncel from Bezmane Vakıf University, Istanbul, Turkey. Thank you for joining my session. Today my talk is going to be about a diversity dynamics analysis of Lassa virus proteome. Let me give you a quick background. Lassa virus is a rodent-borne human pathogen from the Arenaviridae family. It is the etiological agent for hemorrhagic fever, also known as Lassa fever. The disease burden is estimated to be 300,000 to 500,000 with 5,000 deaths annually. The virus is primarily transmitted through the etc. and secretions of infected rodents. There is not an approved vaccine or a therapeutic drug available to date as of July 2021. Considering the risk of Lassa fever endemicity, a better understanding of the structure and function of Lassa virus is necessary. An ongoing aim is to elucidate the proteome sequence diversity from an immunological perspective for novel vaccine therapeutic strategies. Herein, we use a comprehensive quantitative bioinformatics approach to determine the human and rodent Lassa virus proteome sequence diversity. Let me now give an overview of the methodology workflow. In June 2021, all reported Lassa virus protein sequences of human and rodent hosts and their related metadata were downloaded from the publicly available database NCBI virus. Subsequently, identical sequences in the dataset were removed by use of CD-HIT to minimize bias. From all sequences, only 22% of the collected data was found to be non-redundant. Each of the proteins was then aligned by the use of a multiple sequence alignment tool class Omega. Each alignment was manually checked and fixed for any misalignments. Lassa virus sequences were separated according to the host, rodents, and humans. DIMA, a standalone command line tool, was used to calculate channel entropy for each monomer position in the proteome. The chimer size of 9 was used for immunological applications. The distinct peptides at each monomer position were further classified into 4 diversity motifs based on their incidence: index, major, minor, and unique. Now let's go through the results. A total of 3,327 protein sequences were retrieved, with 2,599 and 728 for human and rodent hosts respectively. Figure 1 here provides the Lassa virus entropy plot and incidence of total variance. Let me explain. Entropy values are on the left y-axis, whereas total variance are on the right y-axis. Higher entropy values indicate higher diversity, and the lowest entropy value of 0 indicates complete conservation. Total variance is defined as simply 100% minus the incidence or frequency of the most predominant sequence, which we refer to as index. For example, if a position has a predominant sequence or index with an incidence of 90%, then the total variance incidence in that position is going to be 10%. Human Lassa virus proteome-wide entropy was generally higher compared to rodent Lassa virus. Proteome-wide average entropy value was recorded as 1.66 and 1.30 for the human and rodent viruses. The highest individual protein entropy was 6.61 with matrix protein and 4.25 with also matrix protein for the human and rodent viruses respectively. For a benchmark, the highest monomer entropy value that we have observed from our previous analysis of viruses is that of HIV clade B. The peak absolute entropy of 9.2 and total variance of 98%. Figure 2 provides a scatter plot to better understand the relationship between entropy and total variance. Each dot represents a monomer position. A positive correlation was observed for both human and rodent viruses, with a wider spread in total variance with increasing entropy or diversity, particularly from entropy value of 1 onwards. So an entropy value can have a range of total variance. Given this, we then went to investigate the diversity motifs that comprise the total variance, namely the major, minor, unique variants that I mentioned earlier. Figure 3 provides a distribution for each of these motifs. Each variant, major, minor, and unique, had their own characteristic pattern for each host. The interrelationship between the diversity motifs were complex. In these plots, the x-axis is total variance and the y-axis is incidence. The incidence of that predominant sequence index ranged from 100%, which means completely conserved, to as low as 7.14% for human hosts and 12.12% for rodent hosts. Major variant, which is the second most predominant sequence at a given monomer position, showed a restrictive pyramidal pattern with a few outliers at the end. The peak incidence of 47.40% for human hosts and 58.82% for rodent hosts. Notably, minor variants, the monomer sequence that were absorbed more than once but of lesser frequency than the major variant, appear to be the dominant motif for both proteomes, especially after 50% total variants. While unique variants, which are Slington monomer sequences or observed only once in the alignment, were present at most of the positions with non-zero total variants, with maximum incidence of 58.82% for human hosts and 30.30% for rodent hosts. This suggests that the unique variants are unlikely an outcome of sequencing error. Monotypes evaluate multiple distinct variants with a maximum incidence of 71.42 for human and 44.44 for rodent. The average monotype incidence was higher in rodent viruses compared to human viruses. The violin plots here in figure 3 shows the distribution of the incidence values for each motif. In human Lassa virus, 115 monomer sequences showed complete conservation, which means index incidence was 100%. This study provides an insight into Lassa virus proteomic diversity, fitness change of amino acid, and evolutionary conservation of the virus. Additionally, the reported completely conserved sequences in this study may be attractive targets for vaccine design against Lassa virus. Thank you for attending this talk.